Hello everyone. Today we're going to read a new look at the architecture and dynamics of the Hydra nerve net by Athena Karamidiotti, Sandra Schneid, Christina Boos, Christoph Kramer von Lau, Bianca Bertla, Willi Slaufen-Moser, Martin Hiss, Olga Alexandrova, Christina Glauber, Robert E. Steele, Bert Hoppemeyer, Thomas Holstein, and Charles N. David. Abstract. The Hydra nervous system is the paradigm of a simple nerve net. Nerve cells in Hydra, as in many cnidarian polyps, are organized in a nerve net extending throughout the body column. This nerve net is required for control of spontaneous behavior. Elimination of nerve cells leads to polyps that do not move and are incapable of capturing and ingesting prey. We have re-examined the structure of the Hydra nerve net by immunostaining fixed polyps with a novel antibody that stains all nerve cells in Hydra. Confocal imaging shows that there are two distinct nerve nets, one in the ectoderm and one in the endoderm, with the unexpected absence of nerve cells in the endoderm of the tentacles. The nerve nets in the ectoderm and endoderm do not contact each other. High-resolution TEM and serial block-phase SEM show that the nerve nets consist of bundles of parallel, overlapping neurites. Results from transgenic lines show that neurite bundles include different neural circuits, and hence that neurites and bundles require circuit-specific recognition. Nerve cell-specific connexins indicate that gap junctions can provide the specificity. The occurrence of bundles of neurites supports a model for continuous growth and differentiation of the nerve net by lateral addition of new nerve cells to the existing net. This model was confirmed by tracking newly differentiated nerve cells. Introduction. Nerve cells arose early in the evolution of multicellular animals to sense the environment and control the activity of downstream effector cells. In non-bilaterians, cnidarians, and ctenophores, nerve cells are organized in nerve nets, which are often associated with muscle processes and coordinate the behaviors that involve ectodermal and endodermal tissue. In bilaterian and metazoans, the number of nerve cells is commonly much higher than in cnidarians and ctenophores, and, importantly, nerves are organized into a centralized nervous system with large ganglia. The relatively simple organization of nervous tissue in non-bilaterians offers the possibility of achieving a comprehensive understanding of the relationship between the architecture and activity of the nervous system and animal behavior. With a nervous system consisting of only 500 to 2,000 nerve cells, depending on animal size, the freshwater cnidarian polyp Hydra is a particularly useful model organism for such investigations. Furthermore, there are only two types of nerve cells in Hydra based on morphology: ganglion cells and sensory cells, and there are no surrounding support cells, such as glial cells in bilaterians. Ganglion cells are bipolar or multipolar, but there is no evidence for a distinction between axons and dendrites. Sensory nerve cells, by comparison, are polarized, with a short neurite and sensory cilium at one pole and a longer neurite at the opposite pole. Sensory cells in the ectoderm extend the cilium to the surface of the epithelium to sense the external environment. Sensory cells in the endoderm extend the cilium into the gastric cavity. Recent single-cell RNA-seq results have refined these morphological classes to include 3 sensory and 5 ganglion nerve cell populations based on distinct transcriptomes. Nerve cells in Hydra are organized in a nerve net, as first described in classic studies, in which the nerves are visualized by methylene blue staining and tissue maceration. Although higher concentrations of nerve cells were found in the head and the foot, there was no evidence for ganglia or a brain. A more detailed description of the organization and formation of distinct subpopulations of nerve cells in the various body parts of Hydra polyps was achieved by using monoclonal antibodies that recognized unknown antigens and antibodies that recognized neuropeptides. More recently, the use of in situ hybridization to detect the expression of neuropeptide genes and transgenic reporter lines using the promoters from neuropeptide genes have provided more understanding of the regionalization of the peptidergic part of Hydra's nervous system. However, all of these results lack the resolution to describe the entire nerve net in detail, and there is presently no clear understanding of the overall organization of cells in the nerve net or how the cells communicate with each other. Like Likewise, while extensive EM studies have provided high-resolution images of individual nerve cells, neurites, and synapses, they could not reveal the overall organization of the nerve net. The Hydra nerve net is unique in that it grows continuously. All Hydra tissue, including the nerve net, are newly formed every 2 to 3 weeks in well-fed animals. Since cell proliferation occurs exclusively in the body column, there is continuous displacement of cells from the body column toward the head and foot, and loss of cells at the extremities. In the course of this growth and displacement, the nerve net expands by the addition of nerve cells, which arise by differentiation from interstitial stem cells in the body column. Head and foot specific nerve cells are also continuously added to these differentiated tissues. For example, tentacle tissue is completely renewed in 4 to 5 days by displacement of epithelial tissue from the body. Column into the tentacles. Ectodermal epithelial cells at the base of tentacles differentiate into battery cells with the incorporation of nematocytes and newly differentiated differentiated sensory nerve cells. In the present experiments, we take advantage of a newly developed pan-neuronal antibody, PNAB, which stains all nerve cells and nerve processes in fixed Hydra polyps. Antibody staining and confocal microscopy show that nerve cells in Hydra are organized as 2 separate nerve nets, one in the ectoderm and one in the endoderm. Transmission electron microscopy and block phase scanning electron microscopy demonstrate that the two nerve nets consist of discrete bundles of overlapping nerve cell processes. The bundles include different nerve cell types and neural circuits. Cell type specific expression of annexins and formation of gap junctions provides the necessary circuit specificity. Strikingly, these neurite bundles extend past nerve cell bodies and do not appear to terminate on them. These results suggest a novel model growth of the nerve net based on lateral addition of new nerve cells to the existing net. We confirm this model by tracking the addition of newly differentiated nerve cells to the nerve net. Alright, results. A new peptide antibody stains the entire nerve net in Hydra. A rabbit antibody made against the peptide sequence VTRNQQDQQENRFSNQ. See materials and methods. Stains the nerve net in Hydra and is referred to here as pan-neuronal antibody, PNAB. Figures 1 to 3. In order to determine whether PNAB recognizes all nerve cells in Hydra, 2 sets of experiments were performed. First, we investigated transgenic strains of Hydra, which express neon green or GFP in nerve cells. One transgenic strain uses the promoter of an alpha-tubulin gene, driving expression of neon green. This transgene has been demonstrated to be strongly expressed in all differentiated nerve cells in Hydra, and correspondingly the entire nervous system is brightly neon green positive. Fixed polyps of the strain were stained with PNAB, and transgenic neon green expressing nerve cells were scored for PNAB staining. All neon green expressing nerve cells, 684 out of 684, were stained with PNAB. We also scored smaller number of PNAB stained nerve cells in 2 additional transgenic lines, i.e. HIM176B and N green. See materials and methods. In the HIM176B line, 149 out of 149 transgenic nerve cells were stained with PNAB. In the N green line, 51 out of 51 transgenic nerve cells were stained with PNAB. We conclude from these results that PNAB stains all nerve cells in Hydra and hence permits imaging of the entire nerve net. In the second set of experiments, we compared the number of PNAB stained nerve cells in confocal image stacks with the number of nerve cells determined by maceration of the same tissue. See materials and methods. Both measurements yielded a ratio of 0.12 nerve cells per epithelial cell. Supplemental figure 2C, confirming that PNAB is a pan-neuronal marker staining all nerve cells in Hydra. Imaging the Hydra nerve net with PNAB. Figure 1A shows a low magnification confocal image of the upper body column of Hydra stained with PNAB, including the hypostome and tentacles. The image shows primarily the ectodermal nerve net. Endodermal nerve cells lie below the focal plane. The antibody stains both nerve nets, cell bodies, and nerve cell processes, which are oriented parallel to the oral-aboral axis of the body column. also extend into the tentacles. The hypostome forms a dome above the tentacle ring and includes nerve cells oriented toward the mouth opening. In the center of the hypostome, Figure 1b shows a confocal image of the lower half of the body column. Ectodermal nerve processes are oriented along the oral-laboral axis, but become less oriented in the peduncle. There is an increase in nerve cell numbers in a band just above the basal disc. Confocal images of inverse watermelon polyps in supplemental Figure 1 show the tissue structure in which the nerve net is embedded. Figures 2a and b show a ring cut from the body column in the midgastric region with the ectodermal and endodermal nerve nets. The view is down the oral-laboral axis, and the position of the mesoglea is shown as a green line for orientation. The ectodermal nerve net consists of ganglion cells oriented along the oral-laboral axis, and hence cut in cross-section. The endodermal nerve net consists of roughly equal numbers of sensory nerve cells and ganglion cells. The ganglion cells form a network near the base of endodermal cells, whereas the sensory cells extend prominently up through the endodermal layer into the gastric cavity. Figures 2c and d show images from a single confocal stack taken through the body wall of the gastric region. Nerve cells in the ectoderm form an array of parallel processes oriented along the oral-laboral axis. Those in the endoderm form a roughly polygonal array, Figure 2d. Both nerve cell bodies and nerve cell processes stain with the B and AB. The nerve cell bodies are roughly 10 micrometers long. The distance between nerve cell bodies is variable in both ectoderm and endoderm, but usually 50 to 100 micrometers, while cell bodies and processes in the ectoderm are organized in a plane, which includes the muscle processes of the ectodermal cells. Those in the endoderm are not closely associated with the endodermal muscle processes. See below. Endodermal sensory cells extend deeper into the tissue. See Figure 2b and are not included in the image. Higher magnification images of the hypostoma tentacle and the peduncle basal disc reveal specific neural patterns with a higher density of nerve cells. Figure 3b has a surface view of the hypostome, showing a dense network of ganglion cells with nerve cell processes extending down the body column. Figure 3c shows a section deeper into the hypostome, roughly passing through the mouth opening. Sensory cells are prominent in the ectoderm near the mouth and extend to the outer surface of the tissue. Figure 3b and c, red arrows. When viewed from above, these sensory nerve cells form a ring around the mouth opening. Sensory cells in the endoderm, Figure 3c, yellow arrows, lie between the dense mass of mucus cells and endodermal epithelial cells, which make up the endodermal layer of the hypostome. Supplemental Figure 1a and b. They are connected to a network of ganglion cells in the endoderm under the mesoglea. Figure 3e shows a surface view of a portion of an extended tentacle. Nerve cells form a roughly polygonal pattern associated with the battery cells, which form the ectoderm in tentacles. Single sensory nerve cells, NP1 positive, are integrated within battery cell complexes. In the peduncle, Figure 3f, there is a broad band of increased nerve cell density, which is formed by a unique population of unusually large ganglion nerve cells. These cells sustain strongly with B and AB and have been shown to express gap junction protein, indexin 2, and the neuropeptides M176A and RFA/A. I think that's supposed to be RF alpha. The nerve nets in the ectoderm and endoderm are separate structures and are differently associated with muscle processes. While the images in Figure 1 through B describe the complete nerve net, they do not permit localization of nerve cells or portions of the nerve net clearly to the ectoderm or the endoderm. To resolve this question, we use transgenic lines expressing life-actin GFP, which binds specifically to filamentous actin, since most filamentous actin is concentrated in the muscle processes at the base of epithelial cells. These structures are brightly... present in confocal images. The muscle processes are localized at the base of epithelial cells directly opposed to the mesoglia. Thus, they precisely mark the boundary between ectoderm and mesoglia, and between the endoderm and mesoglia. Figure 4 shows segments of rings of body column tissue. Figure 2a, cut from an ectodermal life-act animal and from an endodermal and life-act animal. Figure 4b. In Figure 4a, the green spots represent muscle fibers in the ectoderm, which run parallel to the oral axis of Hydra, and were cut when the ring was excised from the body column. The irregular red dots in between muscle fibers in the ectoderm are nerve cell processes. They are typically closely associated with muscle processes in the ectoderm. Underneath the ectodermal muscle processes, there is a black space representing the mesoglia, which separates the ectoderm from the endoderm. Red PNAB stains nerve cell bodies and neurites are also present in the endoderm. Figure 4b shows the opposite situation in an endodermal life-act animal. Muscle processes at the base of endodermal epithelial cells are oriented circumferentially around the body column and mark the endodermal side of the mesoglia. An endodermal ganglion cell is stained by PNAB, and neurites from the cell extend parallel to, but are separated from, the muscle fibers. Thus, there is a clear difference in the way the ectodermal and endodermal nerve nets are associated with muscle processes. The diagram in 4c shows this schematically. The localization of ectodermal ganglion cells completely overlaps the localization of ectodermal muscle processes. By contrast, the localization of endodermal ganglion cells is largely separated from endodermal muscle processes. Importantly, in Figure 4a and 4b, there are no connections between nerve cells in the ectoderm and nerve cells in the endoderm. To search more extensively for nerve cell contacts between the ectoderm and endoderm, we image complete rings of body column tissue, Figure 2b, from 4 different PNAB-stained life-act polyps. These rings stained a total of 133 nerve cells, 57 ectoderm, 76 endoderm, and associated neurites, and represented about 4% of the body column tissue. No contacts between the ectodermal and endodermal nerve nets were observed. We have taken many more image stacks at different positions in the body column, peduncle, and epistome, and never found a connection between the ectodermal and endodermal nerve nets. There are no nerve cells in the endoderm of tentacles. In his original description of the Hydra nerve net, Hasti, 1909, noted that nerve cells were not present in the endoderm of tentacles. We have confirmed this unexpected result using PNAB staining of life-act animals. Figure 5a shows an image of the ectoderm at the base of the tentacles in an ectodermal life-act animal. The PNAB-stained nerve net lies above the green muscle processes of the ectodermal epithelial cells. The muscle processes are oriented parallel to the oral axis of the polyp and extend into the base of the tentacle. An image taken deeper in the tissue, below the ectodermal muscle processes, reveals PNAB-stained nerve cells in the endoderm of the body column, but no nerve cells in the endoderm of the tentacles. We also made image stacks through tentacles of the endodermal life-act animals, stained with PNAB. Figure 5c and d. There are many red nerve cells in the ectoderm above the green muscle processes, Figure 5c, but no nerve cells in the endoderm below the muscle processes, Figure 5d. The absence of neurons in the tentacle endoderm indicates a massive loss of endodermal nerve cells during formation of tentacle tissue from tissue of the upper body column. We suggest that this loss of endodermal neurons is related to the formation of battery cells in the ectoderm and reflects a remodeling of both the ectoderm and endoderm during tentacle formation. Nerve cell numbers in the ectoderm and endoderm along the body column. Images of the nerve net in Figure 1 through 3 show only small differences in the apparent density of nerve cells along the body column. To put this qualitative observation on a quantitative basis, we counted nerve cells and epithelial cells in confocal image stacks throughout the body wall at various positions along the body column, from the hypostome and tentacles to the basal disc. The results are shown in Figure 6. Most striking Strikingly, there were no nerve cells in the endoderm of tentacles as seen above. Similarly, the basal disc had very few nerve cells in the endoderm. In contrast to these two differentiated structures, the density of nerve cells in the body column endoderm is relatively constant, 0.1 to 0.19, from the hypostome to the peduncle. The density of nerve cells in the ectoderm, however, changes drastically along the body column, while the ratio of nerve to epithelial cells is relatively constant, 0.15 to 0.22. In the gastric region and the peduncle, the number of nerve cells in the ectoderm rises sharply in the hypostome, tentacle, and basal disc. These are regions known to have a high level of new nerve cell differentiation. See discussion. The nerve net consists of closely associated parallel tracts of neurites, parentheses, bundles. The images in Figure 1 through 3 do not reveal how the nerve net is constructed, i.e. how the size and shape of individual nerve cells contribute to the overall structure of the nerve net. Do the connections between nerve cells and the nerve net consist of overlapping neurites from neighboring cells, or single cell or single neurites, extending from one cell and synapsing onto the next cell to acquire such information? We stain transgenic and green polyps with PNAB. This strain is mosaic, in which not all stem cell precursors are transgenic. As a consequence, in a population of N-green animals, some polyps contain more and some fewer GFP-labeled nerve cells. We took advantage of this mosaicism to identify animals with relatively few GFP-labeled nerve cells. This allowed us to find isolated GFP-labeled nerve cells, and thus image how such an individual nerve cell contributes to the PNAB-stained nerve net. Figure 7 shows two GFP-labeled nerve cells in the ectoderm, with long neurites extending parallel to the body axis. The N-green neurites, Figure 7a, are so closely associated with red PNAB-stained neurites, Figure 7b, that it is almost impossible to distinguish the two. Only occasionally were the green or red neurites locally separated from each other, so that both were visible, Figure 7c. This showed that over long stretches of the nerve net consists of bundles of neurites running parallel to each other. The occurrence of bundles of parallel neurites suggests that a neurite from one cell may not extend along the neurite of a neighboring cell, but also past the body of that neighboring cell. We have found numerous examples of such cell bodies in high-resolution confocal images. Figure 8 shows an example in which a red PNAB-stained neurite passes next to a green GFP-labeled nerve cell body. A single optical section, Figure 8D through F, shows clearly that both the GFP-labeled and unlabeled neurites are stained with PNAB. We also found examples in PNAB-stained preparations in which several tracts of neurites passed over a nerve cell body. In contrast to the above situation, we found few examples of neurites ending on the cell body of a neighboring cell. The ends of GFP-labeled neurites usually lie somewhere along the nerve net at a site that was otherwise unremarkable. See asterisks in Figure 7c. Thus, communication along the nerve net appears to be a case of cell-to-cell communication by neurites interacting laterally to transmit a signal. Section electron microscopy confirms the widespread occurrence of neurite bundles. We sought to confirm the unexpected and widespread occurrence of neurite bundles by transmission electron microscopy, TEM. Hydra polyps were fixed and stained for TEM and sectioned perpendicular to the body axis. Figure 9a is a low magnification image of the ectoderm in the middle of the body column, showing both the apical surface and the basal muscle fibers. Green of the epithelial cells lying directly over the mesoglia, yellow. The muscle processes are cut in a cross-section and form a continuous mat immediately adjacent to the mesoglia. A bundle of 4 neurites lies next to a nerve cell body, NCB red, and a second bundle with 3 neurites lies to the left of the nerve cell body. Both neurite bundles are directly adjacent to muscle processes in agreement with the PNAB stained image in Figure 4a. Figure 9b shows a higher magnification image of another bundle of 4 neurites in the ectoderm. The neurites contain numerous darkly stained glycogen granules and dense core vesicles, which are typically found in nerve cells. In addition, we use TEM cross-section in the mid-body column to measure the frequency of neurite bundles, with different numbers of neurites in a bundle. In the ectodermal nerve net, only about 20% of the cross-section's nerve tracts occurred as single neurites. Figure 9c. About 80% of the nerve tracts occurred as bundles, many as bundles of 2 neurites, but often also as bundles consisting of 3 to 5 neurites. Figure 9c. Figure 9d shows a TEM cross-section of the endoderm in the middle of the body column, with the basal muscle processes directly adjacent to the mesoglea. 2 major differences in the nervous system architecture were obvious when endoderm was compared with ectoderm. First, endodermal neurites and neurite bundles containing numerous dense core vesicles were located at some distance from the endodermal muscle processes. Figure 9D 1, equivalent to the findings obtained with PNAB staining and confocal images. Figure 4b. Secondly, about half of the endodermal nerve tracts were single neurites. The other were bundles containing 2 neurites. Figure 9c. Bundles with more than 2 neurites were not found in the ectoderm. Thus, in summary, the ectodermal nerve net is more directly associated with muscle processes and clearly exhibits higher connectivity than the endodermal nerve net. To extend the TEM results, we analyzed 2,000 serial sections of the ectodermal epithelium in the mid-body region. Serial sectioning was carried out with the block phase SEM method. Figure 10a shows a 3D reconstruction of the serial sections. The image block is 56 by 40 by 13 micrometers and includes 2 nerve cell bodies and corresponding parallel tracks of neurites, purple and green, lying adjacent to muscle processes of the ectodermal epithelial cells. Neurites from 3 additional nerve cells, turquoise, yellow, and red, whose cell bodies lie outside the sectioned block run parallel to and are closely associated with the neurites from the 2 nerve cells shown. The 3D reconstruction confirms at high resolution that nerve tracts between 2 nerve cell bodies consist of bundles of 2 or more tightly associated nerve cell processes. The distance between the 2 nerve cell bodies along the oral-laboral axis is roughly 50 micrometers, in agreement with the distribution of nerve cell bodies shown in Figure 2c and supplemental Figure 2a. A striking feature of the reconstruction in Figure 10a are neurites which run past the 2 nerve cell bodies, blue, purple, and green. This result is in agreement with the images in Figure 7, 8, and 9a, which also show neurites running past neighboring nerve cell bodies. While closely attached to these nerve cell bodies, the neurites do not appear to terminate on them. Figure 10b and c are views from above and below the reconstruction, taken from an interactive 3D model, showing only the nerve cells and neurites. Supplemental Figure 4. The blue, green, and purple neurites extend over the entire length of the reconstructed block parallel to the long axis. By comparison, the yellow and red neurites meet in the middle and form thin terminal fingers, which overlap at the contact site for about 8 micrometers. The yellow neurites also form a thin figure, which appears to contact a spur of the purple neurites for about 4 micrometers, Figure 10c. Thus, there are 2 qualitatively different connections between the neurites, short, thin, overlapping regions, which suggest contact, and long parallel tracks, apparently without contact. Excuse me while I get comfortable. The short spur of the purple neurite, Figure 10c, contacts a muscle process, neurite bundles and neural circuits. The occurrence of neurite bundles raises questions of which nerve cell types and nerve circuits are involved. Based on the Hydra single-cell atlas, there is only one ganglion nerve cell population in the endoderm, N1, and this population forms the endodermal nerve net. N1 corresponds to the endodermal neural circuit, RP2. By comparison, there are two nerve cell populations, EC1/EC5 and EC3, which form the nerve net in the ectoderm. These two populations correspond to the contraction burst CB and RP1 neural circuits in the ectoderm. To visualize how EC1/EC5 and EC3 nerve cells contribute to nerve cell bundles in the ectodermal nerve net, we stained a HIM176B transgenic line, which expresses GFP in EC1 nerve cells in the hypostome and body column, with PNAB, which stains all nerve cells. Figure 11a shows that HIM176B nerve cells form a continuous nerve net in the hypostome and body column. There are no HIM176B cells in the tentacles. The HIM176B cells are also stained with PNAB. Parallel to the HIM176B nerve net, there are additional nerve cells stained only with PNAB. These are EC3 nerve cells. At higher magnification, it is clear that both EC1 and EC3 nerve cells are closely associated in neurite bundles. Since these ganglion nerve cell populations correspond to two different neural circuits, CB and RB1, neurites in the bundles need to form specific synaptic connections with the corresponding nerve cell type. As shown below, circuit-specific gap junctions provide the necessary specificity. Section neurites and bundles are linked by circuit-specific gap junctions. The TEM and block phase images in Figure 9 and 10 do not have sufficient resolution to identify chemical synapses or gap junctions between neurites and bundles. To determine how neurites and bundles communicate with each other, we use higher resolution TEM images on longitudinal sections of ectoderm. Figure 12a and b show two examples of gap junctions between neurites and bundles. Detecting gap junctions by block phase, SCM, or TEM is, however, difficult, and it is not possible to determine which neural circuits are involved. Alternatively, single-cell RNA-seq results can now be used to detect an exon transcript and reveal which cell types contain them. Inexons have been shown to form gap junctions in Hydra, and a search of an updated single-cell RNA-seq database for Hydra has revealed that the two nerve cell populations constituting the CB and RP1 circuits in the ectoderm each contain distinct inexon transcripts. The corresponding UMAPs are shown in Figure 12c. Both EC1 and EC5 nerve cells, which form the CB neural circuit, express annexin 2. EC3 nerve cells, which form the RP1 neural circuit, express annexins 6 and 14, Figure 12c. Hence, the circuit-specific gap junctions can be formed by annexins uniquely expressed in nerve cells of each circuit. Annexin 2 is also expressed in N1 nerve cells, Figure 12c, which form the RP2 circuit in the endoderm. There is, however, no correlation between the neural activity profile of CB and RP2 and RP1 and RP2, indicating that the endodermal and ectodermal neural circuits are not connected. Section nerve net growth occurs by lateral addition of new nerve cells to bundles. A unique feature of the Hydra nerve net is that it is a dynamic structure. It grows and is modified continuously. Since adult polyps do not increase in size, excess tissue generated by cell division is displaced into buds and to the end of the body column. Oh my goodness. Tentacle tissue continuously renewed by differentiation of battery cells at base of tentacles. Basal disc tissue is similarly renewed. Cells are lost by slowing from the tips of the tentacles and the center of the basal disc. How new nerve cells are added to the nerve net was not known. The occurrence of bundles, however, suggests a simple way to add nerve cells to the net by lateral association. Indeed, the image in Figure 7c of two GFP-labeled nerve cells laterally associated with the PNAB stained nerve net supports a model of nerve net growth by lateral addition. To investigate how new nerve precursors are added to the nerve net, we grafted unlabeled AEP polyps to the transgenic and green polyps. Figure 13a. GFP-labeled nerve cell precursors migrated from the bottom half to the top half of such grafts and differentiated into new nerve cells. To localize the sites of new differentiation relative to the nerve net, we stain the grafts with PNAB on day 1, 2, and 3 after grafting. Figure 13b and c show images of young nerve cells and green, associating with the nerve net red on day 2. Migrating precursors without neurites do not associate with the nerve net, and most differentiating nerve cells with short neurites on day 1 were also not associated with the nerve net, figure 13d. However, by day 2 and 3, most differentiating nerve cells extended neurites along the nerve net in figure 13d. The results suggest that outgrowing neurites actively seek lateral associations with the neurites of the existing nerve net. Alright, we made it to the discussion section. There are two separate nerve nets in Hydra, implications for motor control and contractile activity. PNAB staining has permitted, for the first time, detailed imaging of the entire nerve net in Hydra. The results reveal two separate nerve nets, a net of interconnected nerve cells spread throughout the ectoderm, and a second nerve net in the endoderm. There are no nerve cells in the endoderm of tentacles, figure 5, and very few in the endoderm of the basal disc, figure 6. No connections between the two nerve nets are found based on PNAB staining of ectodermal and endodermal nerve cells in the rings cut from the body column of 4 animals. See results. This is in agreement with earlier TEM observations. It also agrees with neural activity measurements, showing no cross-correlation between firing patterns of the ectodermal CB and RP1 neural circuits and the endodermal RP2 neural circuit. In the ectoderm, nerve cell bodies and nerve cell processes are closely associated with muscle processes at the base of ectodermal epithelial cells, figure 4a and c. TEM images of cross-sections of the body column confirm the close association of nerve cells with muscle processes in the ectoderm. This close association is consistent with direct neural control of body column contractions, see below. The localization of the endodermal nerve net is quite different. Endodermal nerve cells and their processes are located in a diffuse band at some distance from the endodermal muscle processes, figure 4b and c, figure 9d. Furthermore, the polygonal pattern of endodermal nerve cells is completely different from the parallel array of endodermal muscle processes arranged circumferentially around the body column. Both results suggest that the endodermal nerve net is not directly involved in controlling the activity of endodermal muscle processes. Recently, Dupre and Hughes 2017 described the electrical activity in nerve cells in a transgenic stain of Hydra that expresses the calcium reporter GCAM6 in nerve cells. They identified 3 discrete, non-overlapping populations of nerve cells based on activity profiles, CB, RP1, RP2. Cells in each population fire together, and there was no overlap between firing patterns. The RP2 population was located in the endoderm, while the RP1 and CB populations were located in the ectoderm. The activity of the CB networks was directly correlated with rapid contraction of the body column. The activity of RP1 and RP2 networks were associated with slower changes in behavior. Oh, did we get it? Come on. Oh my god. The RP2 population is identical with the endodermal nerve net identified by PNAB staining, figure 2d, figure 4b. Moreover, there are no RP2 nerve cells in the tentacles, in agreement with the absence of endodermal nerve cells in the tentacles cells as assayed by PNAB staining, Figure 5. The observation that the RP2 nerve net is not associated with rapid changes in behavior is consistent with the limited contact between the endodermal nerve net and the endodermal muscle processes. Figure 4b and 9d. However, endodermal ganglion cells do strongly express several neuropeptides, example GLW alpha, and release of such neuropeptides could modulate contractile activity in endodermal muscle processes. In particular, Takahashi et al. 2003 have shown that GLW alpha induces slow contraction of the endoderm and consequently elongation of the body column. Almost there. Two ectodermal circuits, RP1 and CB, correspond to the ectodermal nerve net identified by PNAB staining, Figure 2C and 4A, although the nerve net in the ectoderm appears to be homogeneous. The results of single-cell RNA-seq experiments indicate that it consists of two populations of ganglion cells, which express distinct neuropeptides. EC1 and EC1/5 express the neuropeptide M17-6, while EC3 nerve cells express the neuropeptide GLW alpha. To correlate neural activity with muscle activity, transgenic Hydra that express distinct calcium reporters in ectodermal and ectodermal epithelial cells was made. This enables a detection of muscle activity in each epithelium during fast contraction of the body column initiated by CB nerve cells. The results showed that both ectodermal and endodermal epithelial cells correspond simultaneously to activity of the CB circuit, since there are no nerve cell connections between the ectoderm and endoderm. See above, they postulated that the ectodermal and ectodermal epithelial muscle cells activate endodermal epithelial muscle cells directly via gap junction connections across the mesoglea. This conclusion is consistent with our findings. Nerve cells in the ectoderm are closely associated with ectodermal muscle processes, Figure 4a, 9, and 10. And TEM studies have described both chemical synapses and gap junctions between nerve cells and ectodermal muscle processes. By comparison, our results indicate that such contacts are rare in the endoderm, Figure 4b and 9d, and there are no endodermal neural activity associated with the fast contraction of the body column. Section: the Hydra nerve net consists of bundles of parallel nerve processes linked by circuit-specific gap junctions. Prior to the present results, it was not clear how the nerve net in Hydra is constructed. One possibility was that nerve processes from neighboring cells simply meet and synapse, quote, end-to-end, or that a neurite from one cell synapses on the cell body of a neighboring nerve cell, as is the case of bilateral nervous systems. Figure 7 and 8 show that, surprisingly, this is not the case. The connections between nerve cells along the body column mainly consist of two or more neurites attaching laterally to form bundles in the ectoderm, Figure 9c. The EM images in Figure 9 and 10 confirm that neurites in such bundles are closely associated and show, in addition, that neurites and bundles extend past neighboring nerve cell bodies. Furthermore, the images of the nerve net in transgenic M17-6b polyps, Figure 11, demonstrate that neurites and bundles in the ectoderm belong to two different neural circuits, CB and RP1. Hence, nerve cell-specific connections are required to carry circuit-specific signals. Circuit-specific gap junctions between neurites in bundles, Figure 12a, b, and c, appear to provide the necessary specificity. Furthermore, this result is consistent with the observations of Dupree and Used, 2017, that nerve cells in the CB, RP1, and RP2 circuits fire simultaneously, indicated that they are linked in gap junctions. Earlier TEM studies have identified both chemical synapses and electrical synapses gap junctions between nerve cells and between nerve cells and epithelial cells in Hydra. These synapses occurred at multiple sites along neurites, so-called en passant synapses, but were not associated with neurite bundles. The only previously described occurrence of neurite bundles was a nerve ring surrounding the hypostome in Hydra oligactus. This nerve ring contained 20 to 30 nerve cells, and TEM images of the nerve ring showed that neurites and nerve cell bodies were tightly associated, similar to what is seen in the images in Figure 9 and 10. Section nerve cells and polyp form. The body column of Hydra is a tube formed by two sheets of epithelial cells separated by a basement membrane mesoglia. The tube is closed at the basal end by the basal disc, at the apical end by the mouth. The epithelial cells have muscle processes abutting the mesoglia. The muscle process of endodermal cells are arranged circumferentially around the body column. Figure 4b. Muscle processes of ectodermal cells are arranged longitudinally parallel to the oral-epioral axis. Figure 4a. The combined force of these two muscle sheets gives rise to the tubular shape of the body column. In addition to muscle tension, the shape and behavior of the body column is affected by osmotic pressure. Hydra polyps live in fresh water, and there is a constant influx of water into the cytoplasm of the epithelial cells. This water is transferred to the gastric cavity and periodically voided through the mouth, and potentially through the pore in the center of the basal disc. The nerve cells in the two epithelial layers affect the dynamic behavior of the system. This is clearly seen in the nerve-free Hydra. These animals no longer contact and are nearly motionless due to the absence of ectodermal nerve cells. The body column of nerve-free animals is also distended by the increased osmotic pressure, which cannot be released through the closed mouth. However, regular experimental opening of the closed mouth in nerve-free animals leads to release of the gastric fluid and a return to normal tubular morphology of the body column. Thus, one function of nerve cells in the endoderm is the control of endoderm muscle tension in response to osmotic pressure. Section a role for the endodermal nerve net in feeding? A striking feature of the endodermal nerve net is a large proportion of sensory cells, which constitute more than 50% of all endodermal nerve cells. These cells are oriented toward the gastric cavity, Figure 2b, and have sensory cilia, which extend into the gastric cavity to monitor digestive activity and perhaps also osmotic strength. They are connected to the ganglion network at their basal ends and could induce signals affecting changes in the endodermal muscle processes associated with digestion. Two such events occur following ingestion of prey. First, endodermal muscle processes below the tentacle ring contract locally, quote, neck formation, while endodermal muscles in the body column relax. Together, these events create a closed space in the gastric cavity for digestion of prey. After digestion is complete, slow contraction of endodermal muscle processes restores the normal columnar form of the body column. Recent experiments have now shown that endodermal ganglion cells are responsible for this regulation. These N4 nerve cells fire in response to swelling of the body column, e.g. when a food pellet is present in the gut. Ablation of N4 nerve cells using nitroreductase metronidazole NTR-MTZ system prevents mouth opening and injection of the food pellet, which leads to dramatic bursting of the gut. Expulsion of glass beads used as an artificial food pellet was also significantly delayed in N4 ablated animals. Hence, endodermal ganglion cells are involved in regulating mouth opening and swelling of the body column. The latter is a slower process, which could be caused by diffuse release of neuronal signaling molecules affecting muscle tension in the endodermal muscle tissue. Muscle cells. During digestion, slow waves of radial contraction move along the oral-epioral axis, mixing the contents of the gut. Both neck formation and radial contractions are absent or strongly reduced in nerve-free animals, consisting with the role for the endodermal nerve net in regulating digestion. The complete absence of nerve cells in the endoderm of tentacles, Figure 5, which are not involved in digestion, is consistent with this conclusion. Section a model for the increasing cell numbers are displaced into buds, which develop in the lower body column and into the tentacles and the basal disc, which are terminally differentiated and continuously replaced by tissue from the body column. As a consequence of this tissue growth, the Hydra nerve net undergoes continuous addition of new cells. The addition has been clearly shown in experiments tracking the differentiation of DIL-labeled nerve precursors in live animals, or immigration of BRD-labeled precursors. Nerve cell differentiation occurred throughout the body column and maintained the constant ratio of nerve cells to epithelial cells. The DIL experiments, however, could not visualize how newly differentiated nerve cells attached to the nerve net, since the nerve net was not labeled. The new experiment shown in figure 13, in which the nerve net was imaged with the PNAB antibody, demonstrates that new nerve cells are added laterally to the existing nerve net. Thus, the nerve net is an overlay of individual nerve cells and neurites shown in figure 7 and 8, and this overlay is a consequence of how it grows. Figure 14 summarizes these results. In a simple model for growth of the nerve net, one consequence of this model is that elongating neurites terminate at apparently unspecific positions along a pre-existing neurite track. Figure 7 and 10. A second consequence is that elongating neurites grow past neighboring nerve cell bodies. Figure 9a and 10. Both figures are supported by our results and suggest that the existing nerve net acts as a template for the addition of new nerve cells. Section nerve cell differentiation in the hypostome, tentacles, and peduncle. The results in figure 6 show that the nerve cell to epithelial cell ratio in the endoderm is nearly constant along the body column. As discussed above, this ratio can be maintained by continuous addition of new cells to the nerve net during growth. Similarly, in the ectoderm of the gastric region and the peduncle, the nearly constant ratio of nerve cells to epithelial cells can be maintained by addition of new cells to the existing nerve net. The tentacles, hypostome, and basal disc, however, all contain increased numbers of nerve cells in the ectoderm, figure 6, compared to the body column tissue from which they arise by tissue displacement. This implies new nerve cell differentiation in these tissues. This new differentiation has been well documented in earlier experiments and is presumably associated with additional sensory and motor activities in these tissues. Indeed, a fourth GCAMP6S activity pattern was identified in nerve cells at the base of tentacles, which is associated with movement of the head, nodding, a specific population of sensory nerve cells. Homestretch. NV1 was identified, which are incorporated into the battery cell complex in the ectoderm of tentacles. At the opposite end, in the lower peduncle, a population of nerve cells was identified, which expressed the indexin 2 gene. Elimination of the activity of these gap junctions disrupted contractile activity of the body column. In all of these examples, it is possible to imagine that the lateral addition of new nerve cells to the existing nerve net gives rise to local change in neural activity. Finally, the nerve net is not just modified by addition, as described above, but also by deletion of nerve cells. As shown in figure 5 and 6, there are no nerve cells in the endoderm of the tentacles, even though this tissue arises by displacement of the body column, which contains nerve cells in the endoderm. This implies that nerve cells in the endoderm are removed when endodermal tissue moves into the tentacles. It appears that this occurs by apoptosis, although this has not been observed directly. A similar process presumably removes nerve cells from endodermal tissue, entering the basal disc. It also indicates a constant turnover of existing neuron-neuron and neural epithelial interactions. The molecules involved in the underlying feedback communication for these processes are currently unknown. Alright, materials and methods. Animals. Hydra stains were maintained in laboratory cultures and fed 2 to 3 times per week with Artemia nobili. Hydra medium HM contains 1 millimolar sodium chloride, 1 millimolar calcium chloride, 0.1 millimolar 0.1 millimolar potassium chloride, 0.1 millimolar magnesium sulfate, 0.1 vulgaris pH 7.6. The following strains were used for all experiments: Hydra vulgaris AEP wild type, transgenic stains, watermelon expressing GFP and DS red 2 in ectodermal and endodermal epithelial cells respectively, inverse watermelon expressing DS red 2 and GFP in ectodermal and endodermal epithelial cells respectively, and green expressing GFP in nerve cells, a few nests of differentiating nematocytes and gland cells, see below. LifeAct ecto expressing LifeAct peptide GFP fusion protein in ectodermal epithelial cells, LifeAct endo expressing LifeAct peptide GFP in endodermal epithelial cells, Him17-6B expressing GFP in a subpopulation of ectodermal nerve cells under control of the Him17-6B promoter, alpha-tubulin pan-neuronal expressing neon green in all nerve cells under control of an alpha-tubulin promoter. Generation of N-green transgenic line. The N-green line was generated by microinjection of embryos from the SS1 strain of Hydra vulgaris AEP. SS1 is an F1 line that was generated by self-crossing of AEP. The plasmid 5-1 gene bank accession number AY561434, which contains an actin promoter driving expression of GFP, was used for injection. Induction and screening of embryos was carried out as previously described. The N-green line showed GFP expression in all types of nerve cells, a few nests of differentiating nematocytes and gland cells. The N-green line used here is a mosaic containing both transgenic and non-transgenic interstitial stem cells. As a consequence, not all differentiated cells express GFP. Counting cells by tissue maceration. Tissue maceration to identify and count Hydra cell types was carried out as previously described with modifications. Maceration solution is composed of glycerol, glacial acetic acid, and water, 1:1:13. Whole Hydra or tissue pieces were incubated in maceration solution for 10 minutes with gentle shaking to disassociate the tissue. An equal volume of 8% PFA was added to fix the macerated cells. 50 microliters of the suspension of macerated cells were spread evenly over about 2 centimeters squared on a submicroscope slide and dried. 2 drops of glycerol to water 1:1 were added to the dried spread and covered with a cover slip. Nerve cells and epithelial cells were counted under a phase contrast microscope. Counting nerve cells and epithelial cells in confocal image stacks. To count ectodermal and endodermal epithelial cells in confocal stacks, we use transgenic animals expressing GFP in the ectoderm and DS red 2 in the endoderm watermelon line, or vice versa, inverse watermelon line. Since both DS red 2 and GFP are partially concentrated in the nuclei of transgenic epithelial cells, nuclei of epithelial cells can be distinguished from the DAPI-stained nuclei of other cells. GFP nuclei are turquoise, and DS red nuclei are magenta. See supplemental figure 1. The nuclei of all other cells are blue DAPI. See supplemental figure 2b, which shows a confocal image stack in the ectoderm of transgenic watermelon Hydra, expressing GFP in ectodermal epithelial cells. The epithelial cell nuclei are clearly visible as round spots lying above the ectodermal muscle fibers, oriented parallel to the body axis. Nerve cells stained with PNAB in the same image stack are shown in supplemental figure 2a. Yellow spots indicate nerve cell nuclei confirmed by DAPI staining. Preparation and specificity of the PNAB antibody. A rapid antibody was generated against the peptide sequence VTRNQQDQQENRFSNQ, which corresponds to amino acids 4,372 to 4,388 in the cytoplasmic domain of the classic adherent of Hydra vulgaris. Oh boy. Magnapapillata strain 105, GenBank expression number XB01255A722. Peptide synthesis, immunization of 3 rabbits, and subsequent isolation and purification of the IgG fraction were carried out by Dr. J. Pinetta Antibody Service in Berlin. The resulting antibody was highly specific for nerve cells in Hydra vulgaris. Reabsorption with the synthetic peptide blocked the antibody staining. Although PNA peptide in the classic cadherin of Hydra vulgaris, it is now clear that it does not recognize the cadherin protein. Both Hydra alacactus and Hydra viridissima have classic cadherin proteins and nerve cells, and both of these species also strain strongly with PNAB. However, newly available cadherin gene sequences from Hydra alacactus and Hydra viridissima show that the sequence of the intracellular domain of the classic cadherin differ dramatically between the three species, supplemental figure 3. In particular, the 18 amino acid sequence used to make the PNAB antibody is only present in the Hydra vulgaris cadherin, but not in the Hydra alacactus and Hydra viridissima cadherins. Hence, it is clear that PNAB cannot be staining cadherin in neurons, and the identity of the PNAB antigen is presently unknown. Nevertheless, PNAB is a highly specific reagent to identify nerve cells in all species of Hydra. Immunofluorescent staining of fixed Hydra. Immunofluorescent staining was performed based on protocol optimized for double labeling experiments with other polyclonal antibodies. Prior to fixation, animals were relaxed with 2% urethane in HM for a few minutes to prevent excessive contraction induced by fixation. Animals were fixed for 1 hour in 4% paraformaldehyde in PBS, 120 millimolar, sodium chloride 50 millimolar, potassium phosphate 12 millimolar, sodium phosphate, pH 7.2, 7.4. Following fixation, animals were washed 3 times in PBS. Animals used in Figure 8 were fixed with Lewandowski's fixative, 48% ethanol, 3.6% formaldehyde, and 3% acetic acid in water. To improve antibody access to all cells in the polyp, fixed animals were cut in half or in smaller pieces, depending on the experiment. Cutting animals also enabled better mounting of body parts for imaging, e.g. top view of the hypostome or horizontal rings of the body column, allowing views down the y-axis. Cutting was performed after fixation in order to maintain the structural features of the tissue, which was important for analysis of neuronal network characteristics. Immunostaining was performed according to the following protocol. Fixed tissue was watched 3 times in PBS, permeabilized in 0.5% tritone in PBS for 15 minutes, blocked for 20 minutes in 10 mg per ml bovine serum albumin, BSA. 0.1% tritone in PBS. Tissue pieces were incubated in primary antibody and blocking solution overnight at 4 degrees Celsius, then washed 3 times in PBS, and incubated in secondary antibody and blocking solution for 2 hours. Tissue fragments were then washed 3 times in PBS for 10 minutes, stained with DAPI 1 mg per ml in PBS for 10 minutes, and washed 1 time in PBS. Stained specimens were mounted on microscope slides with Vector Shield hearts at anti-vein mounting medium. Beeswax was used on the edges of cover slips to prevent squashing of the preparations. Cover slips were sealed with nail polish and slides were stored at 4 degrees. Primary and secondary antibodies used for staining, together with their respective dilutions, are listed below. Primary antibodies: PNAB Rapid 1 to 1000, Thomas Holstein, Heidelberg, anti-GFP Chicken 1 to 500, Avies Lab. Secondary antibodies: Alexa 88, Donkey Anti-Chick 1 to 400, Cy3 Donkey Anti-Mouse 1 to 200, Cy3 Donkey Anti-Rabbit 1 to 400. Confocal imaging. Specimens were examined with the Leica SP5 scanning confocal microscope. Image stacks were taken with 10x, 20x, and 63x objective lenses. Images at 10x and 20x magnification were taken every 1 micrometer. Images at 63 magnification were taken every 0.3 micrometers. Images were processed with image J. The plug-in stack room was used to correct a Z-shift and to create RGB stacks. Most images shown are maximum intensity projections of short stacks, 5 to 15 micrometers. transmission electron microscopy TEM, and serial block phase imaging SBFI. Sample preparation was carried out according to a modified protocol of Deering et al. 2010, adapted for Hydra. In brief, Hydra polyps were relaxed with 2% urethane in Hydra medium and immediately fixed with 2.5% glutaraldehyde in HM for 5 minutes, cut in half and further fixed for 2 hours. After washing in cacodylate buffer, samples were post-fixed with ferrocyanide osmium tetroxide, treated with thiohydrocarbazide osmium, and in block stained with uranyl acetate and lead aspartate to enhance contrast. After dehydration with acetone, samples were infiltrated with increasing concentrations of acetone resin and finally embedded in Durkheimian ACM resin, mixture hard. The samples were oriented in small silicon molds and polymerized at 60 degrees Celsius for 48 hours. For TEM, ultra-thin sections were cut with an ultramicrotome UCT using a diamond knife and examined with an energy filter transmission electron microscope Libra 120. Images were acquired using a 2x2K high-speed camera and image SB software. For serial block phase imaging SBFI, the resin block was mounted on an SCM stub with conductive glue trimmed to 0.5 by 0.5 by 0.5 millimeters and coated with gold with a sputter coater. 3D imaging was performed on an FEI Quanta FEG 250 scanning electron microscope with an integrated ultramicrotome. At 2.5 kilovolts, 2,000 planes were cut in 3 30 nanometer steps at a lateral resolution of 20 by 20 nanometers, resulting in a volume of 40.5 by 13 by 60 micrometers. Neurites, nerve cell bodies, and nuclei were segmented and rendered using a Mira 5.6 software. We made it, folks. Acknowledgements. Authors thank Sean McCullough of the Denk Lab at the Max Planck Institute for Neurobiology and Martin Stride for help with block phase sectioning, the Center for Advanced Light Microscopy CLA at the Biozentrum for access to confocal microscopes, Angelika Bodeker for laboratory space and resources, Sigrid Grazerod, Natalie Kolb, and Ann Kuhn for hydroculture, and Herman Rohr and Swat Osbeck for discussions. The images in Figure 11 were generously provided by Amber Luwaji. Other contributions: AKSS and CB and OA did PNAB imaging of hydra tissue. CB did a tissue transplantation and tracking of differentiating nerve processors. CCVL and BB characterized PNAB and did initial imaging. WS and MH did DEM imaging and 3D reconstruction of the block phase sections. KMG and RES made the transgenic hydra strain and green. BH and TWH initiated the project at the Technical University of Darmstadt and made the PNAB. BH, TWH, and CNAD coordinated the research and wrote the paper. Funding. They had funding grants from the Deutsche Forschungsgemeinschaft. Oh no, I can't read that. WSMPH were funded by the Marie Sklodowska-Curie Co-Fund Program, and PH and TWH were funded by grants from the DFG. TWH was funded by grants from the DFG. Alright everyone, thanks for listening.